What if the secret to overcoming one of the world's most common chronic diseases could be monitored by looking directly into the human eye? It sounds like science fiction, but a recent breakthrough using human stem cells has done exactly that, and it successfully reversed diabetes in mice. Let's talk about type 1 diabetes. Type 1 diabetes develops when the immune system destroys the cells in the pancreas that produce insulin. Without these critical cells, the body cannot properly take up glucose from the bloodstream. Without that mechanism, maintaining stable blood sugar levels becomes impossible. For decades, scientists have tried to replace these lost cells, but earlier stem cell approaches often produced inconsistent results. Enter a new breakthrough from scientists in Sweden. Researchers have created a more reliable way to generate insulin-producing cells from human stem cells. But how did they fix the old problems? Previously, stem cells would sometimes develop into unwanted cell types, increasing the risk of complications. Furthermore, the insulin-producing cells they did make often lacked the maturity needed to respond effectively to glucose. To solve this, the researchers refined the culture process, allowing the cells to form three-dimensional clusters on their own. This brilliant approach reduced unwanted cell types and created more mature, uniform cells. When they transplanted these optimized cells into diabetic mice, the animals gradually regained control over their blood sugar levels. And the most fascinating part? The researchers transplanted these cells into the anterior chamber of the eye of the mice. This allowed them to monitor the cells in a minimally invasive way over several months, watching as the cells successfully released insulin and responded to glucose. What does this mean for the future of health and medicine? Here are 3 takeaways from this incredible research. Quality over quantity. By letting the cells form 3D clusters naturally, scientists proved that the cellular environment is everything for generating high-quality insulin cells. Targeting the root cause. Instead of just managing symptoms, this points to cell therapies that replace the exact tissue the immune system destroys. The clinical horizon. Building on this success in mice, the researchers aim to work towards clinical translation to eventually treat human type 1 diabetes. By mastering how stem cells grow into 3D clusters, researchers have created mature insulin cells that can survive, function, and reverse diabetes in animal models. The eye wasn't just a window to the soul, it was a window to the future of regenerative medicine. If you are excited about the future of longevity and regenerative medicine, hit that subscribe button.